In the central dogma of biology, proteins are the gears of life activities. Proteins are tightly related to health. Almost all therapeutics act by modulating proteins. Drug targets are mainly proteins. The proteome encompasses the study of all proteins. To better understand life processes, AI-empowered characterization of proteomes is indispensable. Westlake Omics is committed to making breakthroughs in precision medicine and drug R&D by unwavering focus on critical issues that impact life and health. This version is driven by AI-empowered high-throughput proteomic analysis of minute amounts of clinical specimens and complementary omic technologies, supported by technological innovation and multimodal big data. Each and every team member, solidly grounded in the Womics ethos, works shoulder to shoulder as lifelong learners, dedicated to life sciences and medicine towards the overarching goal of enhancing all human well-being in a wise, motivated, innovative, and collaborative manner.
在这个世界上，每个人的基因都是独此一份的特殊存在，是我之所以是我，而不是其他任何人的独特编码。我们将每个人的基因组都看作一本书，这本书的章节也就是基因片段，讲述着生命体从出生、生长发育到死亡的故事。不幸的是，基因书也会偶尔出现错字、多字或少字。当基因片段出现错误，生命体就会患病，比如血红蛋白病、先天性眼盲、先天性免疫缺陷病等。过去能够无障碍阅读这本基因书就是很了不起的事情了，但时至今日，科学家们已经有办法去纠正书中出现的错误。一种新的基因编辑技术 CRISPR 的出现，正在极大的改进我们修正基因组错误的能力。CRISPR 技术又叫成促规律间隔短回文重复技术，相比早前的 TALEN 和 ZFN 技术，具有更灵活、高精度和低成本的优点。这项技术完成基因编辑的过程得益于3个关键角色，一个是运输系统，负责把编辑工具 Cas9 和 gRNA 送进细胞里；一个叫 gRNA， 它就像 GPS 导航，负责在文字浩瀚如烟的基因书里找到出错的地方，然后规划出路线；另一个叫 Cas9， 又叫 CRISPR 关联蛋白9号，它就像工程师沿着 gRNA 规划的路线抵达出错点，对错误进行校正。目前，科学家们意在寻找最先进的 GPS 导航，能够精准地把最高效的工程师送去细胞中特定区域开展基因维修工作。我们通过这样的方式纠正了基因书中的错误，希望在不远的将来，当这项技术更趋成熟，我们面对遗传病、癌症等重症难症时，将手握更多应对办法。在西湖大学功能基因组学与生物信息学实验室里，有一群科学家，他们正在致力于改进 CRISPR 技术，将 AI 和 CRISPR 相结合，使运输系统更高效，让导航不找偏，培训出最能干的工程师。目前技术正在转化落地，从实验室到市场，将会为人类的健康贡献更多的力量。科学家们提升了刊物基因书的能力，丰富了编辑基因书的工具，期待这项技术更安全高效，为疾病的诊断和治疗献计献策。
基于已有的工程化红细胞技术平台，西湖生物医药专注于多个面向临床治疗的重磅红细胞治疗产品的研发，通过血样提取、研发中心工程化改造、工艺化量产、安全性评价、临床回输等一系列流程，完成靶向特异性疾病的针对性治疗。星光不问赶路人，期待未来通过我们的技术，能够真正使得复杂疾病的治愈不再是难以攻克的难题。研发低治疗成本的药物，为每一个生命带来健康的希望。
Hello everyone, welcome to the Westlake Cell webinar series. My name is Yigong Shi, I'm the president of Westlake University. In the next several months, you'll be hearing 10 webinar series jointly sponsored by the Westlake University and Cell Press. You'll be hearing from 40 exceptional scientists, roughly half from Westlake University, half from the international community.
We're thrilled to be partnering with Westlake University to put together a diverse program of events.
The journal Cell is the flagship journal in life sciences. Collaborating with Cell and the Cell Press is such an important strategy and means forward for Westlake University.
Bringing scientists together and fostering constructive communication is at the core of what we do, because we believe that when scientists share their expertise and experiences, and in turn take on board the perspective of others, science is better and stronger, and that's when the best ideas emerge.
Since the early part of 2020, the world and the world scientific community have been strongly influenced by COVID pandemic. We believe this collaboration between Cell Press and Westlake University will be very important, not only for students and researchers in China, but also for the greater research community in the world.
But while the pandemic has posed challenges, it has also pointed to new ways of doing things, and it has in some ways improved communication. Obviously, it will always be valuable to have opportunities to meet in real life. But we can now learn from each other and about the latest scientific developments without spending hours and days traveling to and from conferences and institutes. New modes of engagement are more flexible, they're more sustainable, affordable, and importantly, they're more inclusive.
I hope to see you all at the first webinar series. I'm sure you will feel your time spent at the webinar series truly exciting and even unforgettable.
Good morning, good afternoon, and good evening to our speakers and attendees. I'm Jiaying Tan from Cell and Cell Press. On behalf of my co-host, Professor Shang Cai, and our colleagues at Cell Press and Westlake University, I would like to welcome you to this inaugural event of the Westlake Cell webinar series. We are thrilled to work together in breaking communication barriers during this challenging time, fostering scientific discussions across the border, and bringing cutting-edge discoveries to you wherever you are. I feel excited, and I hope you do too. Now I'm handing the floor to my co-host, Professor Shang Cai, to introduce the theme and our first speaker of today's webinar.
Thanks, Jiaying. Hello, everyone. As the on-site host, I would like to briefly introduce today's webinar theme. So it is nowadays well known that cancer is not simply a pool of malignant cells proliferating. Actually, cancer cells live in an intricate environment with various components such as endothelial cells, fibroblasts, immune cells, as well as some less characterized novel elements such as microbiota. Various components actually orchestrate into this specific cancer ecosystem. The ability of cancer cells to survive and resist treatment has been largely dependent on the specific cancer ecosystem. So how our in-depth understanding of the cancer ecosystem improves the clinical treatment, and how the cancer environment shape the cancer cell plasticity and the progression are being actively investigated nowadays. And today, we're going to have 3 featured talks covering cancer ecosystem from various perspectives, addressing these emerging questions. And each talk will last 25 minutes with 5 minutes question time. And we'll have a 30 minutes discussion panel after the first talk. So with special delight and great honor, I would like to introduce today's first esteemed speaker, Dr. Elaine Fuchs. Dr. Fuchs is a world-renowned stem cell and cancer biologist. She is currently Rebecca Lansfield Professor of Mammalian Cell Biology and Development at the Rockefeller University. She is HHMI investigator. She's also elected member of National Academy of Science. Dr. Fuchs' research has been pioneering in depicting the molecular portrait of skin development, tissue homeostasis, and human genetic diseases such as cancer. In her early research life, Dr. Fuchs has made extraordinary scientific contributions to our understanding of the intermediate filament's role in the normal epidermal cell behavior and genetic disorders. And she has also made a plethora of fundamental discoveries in the stem cell field in characterizing epidermal stem cell identity fate determination, lineage reconstruction, stem cell multipotency, stem cell niche crosstalk, and the stem cell malignant transformation, almost various aspects of the stem cell biology, and many of which could be written into the textbook. In recent years, much of her work has been focusing on communication of the tissue stem cell and the environment, particularly the immune cells and the various stresses. Due to her exceptional contribution to science, she has won numerous awards. It's such a long list that I couldn't finish reading it today. In addition, she has been a very successful mentor. Many of her trainees have been very famous in their respective scientific field all over the world. In summary, Dr. Fuchs is a scientific pioneer in the field of stem cell, skin stem cell and cancer, and her work is tremendously advancing our understanding of the skin biology and provide us with hopes for disease therapeutics. And today, it's our great pleasure to have her joining us and sharing with us her exciting scientific findings. Without further delay, please join me and welcome Dr. Elaine Fuchs.
Thank you so much. Can everyone hear me? You can hear me?
Clear.
Yes.
Okay,
Very clear.
very good. And you can see my screen. Okay, so let me just start by saying that it's a real honor for me to be opening up this webinar series and to have been invited to give this talk today. I wish I could be there in person. It's been a while since I've been to China now, given the pandemic around the world, but hopefully that will change soon and we'll all get together again. So today I'd like to tell you a little bit about the work that my laboratory has been dealing with regarding stem cells, really in a venue of survival of the fittest. And what better survival of the fittest is there than to start with a series of pictures of a recent trip, not so recent now because of the pandemic, that I took to the Galapagos Islands. And I think it serves as a beautiful example of the incredible diversity that nature has had in creating body surfaces in a way that she just hasn't done this for any other tissue of the body. And one of the reasons is that our surface is basically exposed to such various different extremes, environmental extremes and other types of extremes. And in order to be able to really meet these needs, there has to be a tremendous amount of tailoring and evolution going on. So we've just really begun to scratch the surface in our understanding of the skin epithelium. It is indeed our barrier from the outside world, and it's exposed to a variety of different stresses, ranging from mechanical stress, metabolic stress, pathogens, temperature and pH extremes, ultraviolet light, wounds, oncogenic stress, and the tissue stem cells must be able to be poised to be able to sense these changes and rapidly adjust. And this is the basic problem that my laboratory has been focusing on. Over the years, we've identified where the stem cells are in the skin, and we showed that normal skin tissue stem cells always reside at the interface between the epithelium and the mesenchyme, the epidermis and the dermis. And they exist and produce a basement membrane that then separates these two structures. We also know that the epidermis and the sebaceous glands turn over continually, and their stem cells thus must be active all the time. We know that the hair follicles, by contrast, undergo bursts of periodic regeneration and degeneration, and therefore they're only periodically activated. We know that they all have a common embryonic origin, and they share many of the same features, including keratins and certain integrins. But although they have these shared features, they are defined largely in task and their gene expression by their local microenvironment or their niches. We know that normal stem cell niches have intimate associations with the vasculature. And for the hair follicle, it's the lymphatic vasculature. In work that was done within the last year and a half or so, Sherry Cohen in my laboratory used three-dimensional tissue clearing to be able to look at the three-dimensional structure of the skin. And what she found was that, in fact, the lymphatic capillaries are very closely associated with the hair follicle stem cells, as you can see here, the capillaries in red and the stem cells in green. This coordinates the hair follicles across the tissue, and one of the things that must happen is that all tissues must have their stem cells coordinated to be able to perform specific functions. And you can see here that the stem cells in green have this close association. The early activated cells in white don't have that association. So for the hair follicle, the connections are really associated with the quiescent stem cells. And this just shows you the example that all of the stem cells are doing everything similarly at the same time. They're synchronized in this way, and it's the lymphatic connections that allow them to be synchronized. And indeed, if we take a look at how this synchrony works and how the stem cells transition from a quiescent period to an activated period, we know that the stem cells themselves produce lymphatic connectors and lymphatic dissociators. During the quiescent period, we know that the lymphatic connectors are high, and in order to be activated, they must be downregulated. We know that the lymphatic dissociators, such as angiopoietin-like 4, must be high in order to be activated, but during normal quiescence, this factor is low. So there's a lymphangiogenic switch that takes place at the start of stem cell activation and growth. It raises a series of questions about what this connection is doing. Could the transient fluid pressure that builds up right at the activation stage promote proliferation? Could it perhaps, during the quiescent phase, be draining certain types of immune cells, or perhaps draining certain activating growth factors? There are a whole variety of different questions. We also know that there's a signaling switch that occurs as stem cells go from quiescence to activation. BMP and FGF18 signals are high in the quiescent state, and they're downregulated as the cells transition to activation. Conversely, Wnt and sonic hedgehog signaling is high in the case of the activated state, but it's low in the quiescent state. We also know that there's a switch with regards to transcription factors. TCF3, TCF4, a whole variety of different transcription factors are high in quiescence, and they're downregulated as the stem cells become activated and undergo their track to lineage determination. And finally, we know that the differentiation transcription factor genes are downregulated, and they are upregulated in order to be activated and march down this pathway. So this has given us quite a bit of information and given us a lot of changes that are taking place as stem cells transition from quiescence to activation. How about the chromatin level? What's going on there? In work done by Renee Adam and Hansel Young, two former graduate students in my laboratory, we studied this problem. We took a look at all the transcription factors that are expressed in the quiescent hair follicle stem cells, and we used ChIP-seq analysis to take a look at where these various factors bind. We looked at whether or not they bind to a common region, and we found that there are quite a few of those common regions where all the different transcription factors are bound in these very short stretches. These stretches are located within large open chromatin domains called superenhancers. They are also marked by certain activating histone marks such as H3K27 acetylation and by mediator components, which are components that are bringing the promoter and enhancer together. And if we hone in on these, we can see that the superenhancer regions are riddled with these high platforms for the binding of all of these factors. Intriguingly, when we take a look not only at the transcription factors, but also the DNA effectors for those signaling pathways that I was talking about, we can see that in the quiescent state, phosphosmad1, a readout of BMP signaling, is bound to these stretches of DNA. And we can see that TCF3 and TCF4, which in this quiescent stem cell context are raising the threshold for Wnt activation, are also bound in these regions. So what that makes these regions are molecular zip codes for who I am, a hair follicle stem cell, and where I am in a quiescent stem cell niche. There are only about 350 genes that are regulated by superenhancers of all of the genes expressed by the hair follicle stem cells, so it's only about 5%. But these are critical genes. They are the transcription factor genes themselves, the BMP and the BMP receptors, the Wnt inhibitors and the Wnt receptors, and so on and so forth, along with a number of skin disease genes. So effectively, these small percentages of genes that are regulated by superenhancers in the hair follicle stem cells turn out to be a rich list of critical genes. We know that stem cells are tuned to their environment, and upon a change, they have to rapidly survive. Stem cell plasticity turns out to be driven by superenhancers. So if we take quiescent stem cells and we put them in culture and we passage them under conditions where there's rich serum and growth factors, the stem cells will divide, but we know that they're still stem cells because we can re-engraft them back onto a mouse and get hair. And interestingly, we also get epidermis and sebaceous glands. Normally, these stem cells in their native context will only make hair follicles, not epidermis or sebaceous glands. So they acquire a broadened lineage specificity when they're passaged in culture under different microenvironmental conditions. Indeed, the superenhancer transcription factors are downregulated in culture. There's only about 100 of the superenhancers that are maintained in culture. Many of the transcription factors associated with quiescence are downregulated in culture, but it turns out that wound-induced transcription factors are activated, and indeed this is mimicking a wound-induced microenvironment. And so by taking the stem cells out of context, inducing them to proliferate, and stimulating them with serum and growth factors, it's as if the quiescent stem cells are responding to a wound where they exit their niche, migrate upward, and re-epithelialize the epidermis. So how can we challenge this and demonstrate functional significance? Here we took these epicenters and we used them as enhancers to drive EGFP in mice. The way in which we make the mice is to take a lentivirus, package this gene, and introduce it into the amniotic sac of a 9.5-day mouse embryo. The embryo takes up the lentivirus only in the first cell layer, which happens to be the layer of unspecified skin progenitors, and then we can allow the mouse to grow up and analyze where GFP is expressed. We see it only in the quiescent stem cells of the hair follicle in their quiescent niche. When we put the cells into culture, the GFP turns off. We can graft the cells back on to the mouse, the GFP turns back on again. In opposite fashion, if we now take an in vitro epicenter and use that to drive EGFP, in the unwounded state, there's no activity. In the wounded state, now the activity turns on. So again, these are really environmental sensors, and superenhancers are environmental sensor enhancers that basically are able to respond to the changes that are happening in the microenvironment and that is experienced by the stem cells. So let me switch now into this issue of what happens when stem cells acquire oncogenic mutations and how do they deviate from their course. Here we've been focusing on squamous cell carcinomas, a non-melanoma skin cancer that is one of the most common cancers worldwide. And it's also, when you consider that squamous cell carcinomas can affect not only the skin, but also the head and neck, vaginal, cervical tissue, esophageal tissue, and many other tissues. It's one of the most deadly cancers worldwide. Even when we look simply at the skin, patients on immunosuppressive drugs or BRAF inhibitors for melanoma, for instance, dramatically increase the risk of metastatic skin squamous cell carcinomas. In 10 years, the incidence of metastatic cutaneous squamous cell carcinomas is predicted to more than double. And as our ozone layer continues to be damaged from global warming, we're getting an increase in skin cancers. So as stem cells accumulate oncogenic mutations, particularly oncogenic mutations that raise the level of RAS-MAP kinase signaling, then cancers can arise, and that drives them to ultimately a squamous cell carcinoma. We were particularly interested then in identifying where are the tumor-initiating stem cells that exist within squamous cell carcinomas. Many years ago now, we took an approach that was basically similar to the approach that we took to find the skin stem cells in the first place, and that is to fractionate the cells, in this case by fluorescence-activated cell sorting, and then sampling the various different pools. We found that nearly at the single cell level, we could take particular cells from our purified populations, introduce them into a host recipient mouse, and get a squamous cell carcinoma like the initial parent. That told us that we had a so-called cancer stem cell, more frequently referred to as a tumor-initiating cell. So what do these things look like? They don't look anything like the normal counterparts. They do have some similarities. Intriguingly, hair follicle stem cells and epidermal stem cells are both vehicles that, when they acquire mutations, can accumulate them and ultimately give rise to squamous cell carcinomas. There are vestiges of both hair follicle stem cells and epidermal stem cells within the molecular signature of these tumor-initiating cells. But there are many differences, including many differences that we already knew about associated with cancers. So what happens on the path to cancer that leads to this dramatic shift in gene expression? In work that Ye-Jing Ge did when she was a postdoctoral fellow in my laboratory, and she's now an assistant professor at the MD Anderson Cancer Center, we learned that cancer is really a wound that never heals. And this is at the molecular level, at the chromatin level. You can look at chromatin landscaping of an epidermal stem cell or a hair follicle stem cell, find very different patterns. As I already mentioned, the wound-induced stem cell pattern is yet different again, and it's similar irrespective of the direction of wounding. But when we look at what happens in the cancer or the tumor-initiating cells, we see a resemblance to the wound-induced pattern, but now additional chromatin peaks opening up. It turns out that these additional chromatin peaks opening up have very rich density in ETS2. ETS2 is interesting because it is phosphorylated by the MAP kinase pathway, which is elevated transiently in a wound and elevated constitutively in squamous cell carcinomas. So we can take these new peaks that open up in cancer and again see whether they have functional significance. In the unwounded state, there's no activity. The wounded state, no activity. And now, during malignant transformation, we see activity. So again, these are reflective then of a changing microenvironment and a changing, in this case, combination of microenvironment and genetics. We can also demonstrate functionality of the ETS2 and its phosphorylation. If we create a phosphomimetic of ETS2 and introduce it in an inducible fashion in the skin what we can see is that when you induce ETS2 in a very short period of time, the tissue rapidly transforms into a malignant-like state relative to the control. And if we look at the super-enhancer landscape, what we find is very similar super-enhancer landscape between the ETS2 phosphomimetic cells and the squamous cell carcinoma stem cells. So again, there are these parallels, and it allows us to be able to use a combination of chromatin landscaping and functional analysis to get at the heart of understanding mechanism. Another interesting aspect of the cancers is that the microenvironment still matters in many of these cancers, if not most of these cancers. We know that the stem cells that I've been talking about, that are the tumor-initiating cells of squamous cell carcinoma, are located at the tumor-stroma interface. But the stroma is very different now. There's cancer-activated fibroblasts, there are immune cells, there are blood vessels, and this dramatically changes the microenvironment that these stem cells experience. We also learned that wherever blood vessels come up next to the developing cancer, that there is an active TGF-beta signal that now signals to these tumor-initiating stem cells. That then causes a change in their behavior, and intriguingly, it is tumor heterogeneity caused by differences in the microenvironment, in this case, the perivasculature. And so we've generated mice that can glow red, whose cells glow red when they receive a TGF-beta signal. And when we do that, we see that the tumor-initiating stem cells at nearby a blood vessel, which would be right here, are now glowing red relative to the stem cells that are further away from the perivasculature. We can also link a Cre recombinase activatable by tamoxifen to the TGF-beta sensing enhancer, and that now allows us to switch the color from red to green, but it also allows us to look at these cells, and we find that they're slower cycling due to TGF-beta being promoting a slow cycling behavior in epithelial stem cells. However, when we lineage trace, we also see that in fact these tumor-initiating cells are invasive, breaking down the basement membrane and invading. And remember that the blood vessel is right here, whereas the tumor-initiating stem cells that are further distant from the blood vessels don't show this kind of activity. They're much more fast in proliferation, but they contain smooth borders. We also then went on to demonstrate that, in fact, this further matters with regards to the biology, because if we take a look at what happens when you treat these tumors with cisplatin or with immunotherapy, the two drugs of choice for treating head and neck cancers, what we find is that within a matter of days, caspase-3 in blue lights up the majority of the tumor. But the lingering red cells, the TGF-beta responding stem cells, still remain. And if we wash away the cisplatin treatment and now activate our lineage tracing marker, we can see that when the tumor grows back, which it invariably does, it becomes green, indicating that it's derived from the TGF-beta responding stem cells. We've also engineered mice that allowed us to do an adoptive cytotoxic T-cell therapy. We created a mouse where the mouse stem cells are expressing OVA. We now add cytotoxic T-cells that can target OVA. There's dramatic infiltration of the cytotoxic T-cells in a short period of time, and not surprisingly, the tumor shrinks. But the tumor never shrinks entirely, and the tumors eventually relapse. And it turns out that they're not killing the TGF-beta responding tumor-initiating stem cells. They go from being less than 2% of the naive tumor to nearly a quarter of the naive tumor within merely a week. So how do the TGF-beta responding tumor stem cells evade cisplatin and immune attack? I won't go through this in detail because we published this already, but there is a parallel between the activation of glutathione genes by these tumor-initiating stem cells, and that correlates with poor prognosis with regards to human head and neck cancers. We also showed that TGF-beta signaling results in the upregulation of CD80, which is a member of the PD-L1 family of ligands, and that at low levels, CD80 couples with CTLA4, and that dampens down cytotoxic T cell activity. And so in this fashion, the tumor cells then are able to escape detection by the cytotoxic T cells. So that then leads us to wonder whether the blood vessels are merely bringing a TGF-beta rich immune cell environment to the tumor stroma interface, or do blood vessels have an impact on tumor stem cell survival? We've looked at this question, and what we've discovered is that tumor stem cells have an altered metabolic diet over their normal tissue counterparts. The tumor cells are in a stressful microenvironment. They downregulate protein translation, and coupled with that is also an altered response to the metabolism. This was one of the top 10 Sloan Kettering cancer science breakthroughs of the year. It was done by one of my former MD PhD students, Sanjay Baksh, along with Lydia Finley, our collaborator at Sloan Kettering. And what we demonstrated was that, in fact, the stem cells become addicted to intracellular serine. They could make serine on their own, but they don't. And they need it in order to be able to survive. So what is happening? How do they become addicted to serine uptake? They do so in order to avoid the production of alpha-ketoglutarate in the serine biosynthetic pathway. And what happens is that alpha-ketoglutarate is a substrate for histone demethylases, including H3K27 demethylation. And so what happens when you put the animals on a serine glycine-free diet is that they downregulate H3K27 methylation, and that leads then to the differentiation of the tumor, as you can see by keratin 10 and as you can see by the staining here. We also showed that a human xenograft of a tongue squamous cell carcinoma and another head and neck squamous cell carcinoma responds similarly. So what I've suggested to you then is that some cancers might be controlled by adjusting the diet. And there we can see, if we put the animals on a serine glycine-free diet, we see that tumor burden is dramatically reduced. We don't yet know whether this would happen also in humans. So what I've shown you is that cancer is really a tango of a whole variety of different environmental stresses that are basically giving rise to survival of the fittest. And what are the prospects for therapies? Perhaps targeting mechanical properties in cancers. I didn't have time to talk about some of our recent work on this subject. RAS-MAP kinase has always been a difficult druggable target. ETS2, though, offers some suggestions. EIF2A really came from our translational studies several years ago. TGF-beta inhibitors could be useful, but only if used in a temporal sequence with other therapeutics, and CD80 immunotherapy, or shifting the animal's diet, or in this case, the human's diet, would be the ideal situation. So these are the people responsible for the work that I talked about today, and hopefully next time I'll be able to tell you more about the work that we've been doing. Thank you again to the organizers and to Self Press for inviting me to give this lecture.
Thank you, Dr. Fuchs, for a very insightful talk. Now we have 5 minutes for question time. I will take the privilege to ask the first question. So, Dr. Fuchs, as you know, that cancer-initiating cells is actually less frequent than the non-cancer stem cells in the tissue. And these cells seem to have very strong ability to avoid being attacked by the drugs and also immune systems, right?
Yes.
And how do those non-cancer stem cells survive in the tumor? As we know that cancer stem cell can survive, evade immune attack, right? Do you think the cancer stem cell protect the non-cancer stem cells in the tumor, or do you think they have other pathways to evade the immune system?
Yeah, you're raising some very interesting possibilities and very interesting suggestions. We've been looking at cellular competition. Initially, we looked at cellular competition in normal skin development, but now we're also interested in various different aspects of cellular competition. Are the normal cells competing with the cancer cells or vice versa? So what's happening before you get to the full-blown cancer? The other aspect, though, that I think is really intriguing, and the area that we're really focusing on coming up with cancer treatments, is to really exploit the differences that exist, the molecular differences between the cancer stem cell and the normal stem cell, because therein lies the roots of how do you develop therapies that will target the cancer stem cell and not target the normal tissue stem cell. And I think some of the avenues that I've brought up today provide those kinds of venues for that possibility. So there's still a lot to be done with regards to understanding these problems, but I think it's a very exciting time in the field.
Okay, thank you.
Elaine, a very beautiful talk. Thanks for joining us at this hotel. So I have a question about your super enhancer as an environmental sensor idea. It's a great one. I think many signaling pathways, obviously the distal end of it is a modified transcription factor. So the question is, are there cis elements within the super enhancers that directly recruit them there, or is it actually they target the promoter, which then via the promoter enhancer loop then get targeted to a super enhancer?
Yeah, great question. We think it's within the super enhancer itself, and the reason for that is this aspect of these DNA effectors for the signaling pathways that are necessary to make a switch. They're right there. And so what we're thinking is, and doing the experiments to really test this hypothesis, is that you have phospho-SMAD, for instance, right there in the middle of the super enhancer regulatory elements. What happens then when you start to add BMP inhibitors? The phospho-SMAD presumably is no longer able to remain there. That then, and if we mutate those sites, we see a loss of super enhancer activity or diminishing super activity. Another interesting aspect is Wnt signaling, because we were surprised to see that in looking at the activated genes that are regulated by Wnt signaling, what we find is that a gene like LEF1, which is necessary for the next step down the road, has TCF3 and TCF4 bound to the exact same site that LEF1 is going to end up, and TCF1 is going to end up binding to. And so what we're operating on the principle is that you need to have beta-catenin there in order to be able to interact with TCF3 and TCF4 to remove it from chromatin in order to let the other active beta-catenin partners be able to get in and bind to the chromatin and activate it. With regards to our footprinting on the super enhancers that are associated with the quiescent state, what we see is just TCF3 and 4. So we think that that's pulled off probably by BMP inhibition and by Wnt signaling together, and that then is what's silencing one super enhancer set and then activating, in that case, the downstream lineage super enhancers. So the mutagenesis that we've done is consistent with that, and the histone and chromatin mapping that we've done is consistent with that.
Great. Yeah, thank you.
Hello, Elaine. This is Yinong Zhou, a former trainee with John Masake, and we have been neighbors for a long time. Yeah. I have a general question related to the stress sensing by the epithelial cells, because you particularly mentioned that this type of the skin stem cells are very sensitive to all different kinds of stress. I'm just wondering at a high level, what do you think about how like a single cell type would be able to respond to various stimuli, but then they may react similarly by stop or activate their proliferation?
Yeah, great question. With regards to one type of stress I can give you is that we were looking at translational control several years ago, and we did a screen for a whole variety of different translational regulators, and we looked at early what happens when a cell gets early one oncogenic hit versus no oncogenic hit. So we did an in vivo screen, and what we found is that the canonical regulators for translational initiation turn out to be essential for wild-type stem cells, but less essential for the cancer stem cells or the oncogenic hit. Whereas conversely, the non-canonical translational regulators turn out to be required for the oncogenically stressed stem cells. And what that told us, and as we went on to look at this, is that translational control is dampened down in when the stem cells receive an oncogenic hit. And associated with that is a dependency now on a non-canonical pathway. And that dependency turns out to allow translation selectively of certain oncogenes that otherwise are not going to be elevated by the normal pathway. And so that's one example of how you end up with essentially the same microenvironment, but now just due to a genetic hit, a dramatic change in translation that then impacts one type of stem cell, in this case the cancer stem cell, and not the other type. And so you end up, both of these translational regulators, EIF2 alpha and EIF2A, are both present in the normal and in the cancer cells. But what happens is that the cancer cells end up getting much higher levels of phosphorylation and decommissioning of the canonical pathway. And exactly how that happens and what the stress is that they're feeling, whether it's sort of being having a foreign cell in the midst of a sea of wild type cells, is there competition coming into play? I think all of these kinds of questions would be really interesting to look at in the future.
All right. Thank you, Dr. Fuchs, for your great talk. Let's move on to the next session. I'll hand it over to Jiaying for our discussion panel.
Thank you. So now we'll move to the panel discussion, and it's my great pleasure to welcome our three panelists. Dr. Elaine Fuchs, who you've just heard from. We also have Dr. Florencia McAllister, a physician scientist with both basic research and clinical experience in tumor immunology and pancreatic cancer. And our third panelist, Dr. Xiang Zhang, who has dedicated his laboratory in studying metastatic breast cancer. Thank you all for coming.
Thank you.
So I see that while you each worked on tumor of a different tissue of origin, right? But your research has all touched upon the interplay between cancer cells and the microenvironment, in particular, the immune environment. So I'm curious, what do you think is the most intriguing aspect of that tumor-non-tumor interplay in your favorite context? Florencia, you want to start?
Yeah, so certainly so first of all, thank you so much. This is wonderful. And just, the talk we just heard was fantastic. In that regards, one of the things that I kept thinking after Dr. Fuchs' talk and in light of your question is, of course, my interest lies in microbes. And, we find all along that there are changes that we all know are very clear in tumors, like, vascularization, changes even in nerves, in fibroblast composition. And we tend to think about those when we think about, structural changes, but microbes and also changing all along in tumor and non-tumoral areas based on these factors. If you have more vascularization, you're going to have more leakage of bacteria from other sites. The same thing with structural barriers that may or may not happen when you have bacteria, and some of their products, as I'm going to mention, nets can end up becoming a barrier to immune cells themselves. So I think, again, it's just kind of as preceding my talk, I do believe that, of course, not everything is microbes, but since that's my main interest, I think that microbes may play a role that may have been not uncovered before, and I think it may be one of the not perhaps the root of the changes, but perhaps one of the factors that help of that progression. And we are finding that along even in sites where there's no phenotypic changes, like in metastatic sites, we are seeing changes that are preceding the seeding of cells. So we are seeing that even prior to seeing premalignant lesions visible to the microscopic view, you already have some differential seeding of bacteria, which might be following some of these changes, perhaps in stem cells, perhaps in other factors associated with perhaps different oxygenation or pH. So I didn't want to talk only about bacteria, but I always end up there.
Bacteria is fascinating, I have to say. Xiao, since we mentioned metastasis and you've done a lot of work, really outstanding work in this area, I'm curious of your thoughts of Florence's point.
Sure. I think that my favorite kind of aspect of this is this multidimensionality of this cancer ecosystem interaction. So as we see a lot of beautiful images from Dr. Fuchs' talk, and that's already kind of showing clearly this is three-dimensional at least, right? And then at time, this cell and the cancer and the niche interactions will evolve. So you have a temporal factor there. In cancer in particular, because cancers are so heterogeneous, even for the same type of cancer, you have a great amount of heterogeneity, not only in cancer cells, but also in microenvironment, right? So there's this cancer microenvironment interaction may vary from patient to patient, from one cancer to another cancer. So put this all together, I think is a major challenge, and of course microbit organism is another thing that may distinguish one patient from others. So all of this put together is a multidimensional question to me, and that's the major challenge, I think. Yeah.
Thank you. Elaine, what's your take on that aspect?
Yeah, I mean, I love the other two answers as well. It's my instincts were to think about some of the recent work of one of my former postdocs, Nogi Oshimori, who's now at Oregon State Health Science Center, and he's been following up on the perivasculature with the TGF-beta. We knew back then that TGF-beta is coming from the monocytes. We had not characterized the monocytes in detail, nor understood exactly how they get there. They're associated with the blood vessels. And what was interesting is that the stem cells themselves, they are expressing interleukin-33, which ends up feeding back to the monocytes and promoting their differentiation to type 2 like macrophage. And so essentially the stem cells are cross-talking with the immune system that's located in the stroma, and we're very interested in how did it start to go along this path that has given it this deviousness at the end point. And so it's certainly all of what we've studied with regards to the immune system and the stem cells. It's a two-way street, and it changes in cancer. Somewhere along the line, the tumor, the stem cell ends up initially getting wiped out if there's an oncogenic hit or the like. It gets cleared out by the immune system, but later on, it doesn't get cleared out. And it's these changes that are happening, and we still don't know the temporal sequence of events, but I think they're going to be very interesting to study in the future. So, and the microbiome, of course, the microbiota, I mean, it's a fascinating question, because certainly the mechanical stresses that are on tumors as they're developing gives rise to all sorts of leakiness in the blood vessels and in the lymphatics that are really that are likely to really change in terms of the pathogens that are coming in. So yeah, a lot to work on.
Thank you. Very insightful answers.
So microbiota is certainly a novel component in the tumor microenvironment in recent years. Dr. McAllister, so do you think this microbiota is actually integrating into this complex, already very complex cancer ecosystem, and exert some role linking the epithelial cells and the immune system, or they have their specific or unique roles specifically to the cancer cells?
Well, first of all, I don't think we know. We know so little about microbes in general in terms of regulation of tumors, but even less about the direct effect of microbes on tumors themselves. I think most of the work in the past sort of decade has been very descriptive and characterization of a lot of various microbiome across different type of tumor types, but it's been mostly related to gut microbiome. The knowledge about the role of microbes within tumors, it is really limited. I would say less than a handful of papers. And again, I think most of those papers are suggesting its importance. And I think most of those labs who have done those papers, and I'll talk a little bit about that today, we are now working on sort of that next step, which is more of the mechanistic understanding. And, if you ask me from the two possibilities that you give me, I would say, always I start by thinking, we try to be reductionist because it's always easier, but things are always a lot more complex than we envision. So I will not think that it's an effect direct on the epithelial cells. And we are already seeing some clues about that it's not only a bacteria, it's always like groups of bacteria. It's not even that group of bacteria, it's more like the function, and it's more like the metabolites. And even those are going to be heterogeneous within patients. They're going to be heterogeneous within tumors, because it's the same tumor. If it's located in the head of the pancreas or the tail of the pancreas, just as an example, certainly it's going to have different nutritional status, different oxygenation status. So there's going to be a lot of heterogeneity found, and I think it's going to be complicated, but I think we are going to learn a lot about this, and I feel like in the next few years, a lot of publications are going to come about that, to answer that.
All right. Thank you. Cancer ecosystem is already very complex, and now we have another layer of complexity. And we know that as we learning more and more about the non-tumor components in this cancer system, so do you guys think, are we going to find any pan-cancer features that can break the boundary of the organ or tissues that can be used for future therapeutic development? Dr. Zhang, what do you think? Do you have any thoughts about this?
I don't have any concrete data to speak to that, but my guess is, and this is also the future direction of my laboratory, is to break this barrier between different cancer types. For instance, we focus on bone metastasis. We are now seeking to understand the common feature of cancer cell, all from all tissue of origins. Is there any common mechanism they use? We already know some. We're going to do more systematically investigation on different types of cancer. How do they colonize bone through different or common mechanisms? And we do think there are common features. For bone is a unique microenvironment, have a lot of unique resident cells. To survive there, you may expect that the selected pressure will select for some convergent traits of the cancer cell. So that's a great question, and that's one of the future directions, at least for my lab. I believe it's also for many other outstanding labs. Yeah.
Okay. Dr. Fuchs, do you have, what's your take on this question?
Yeah, I think back to some of the early work that was done by Beatrice Mintz and Mina Bissell and others, showing that when you take a tumor cell and you put it in a completely normal microenvironment, it really has all of the features of a normal cell, even though it can be very, very genetically altered. So I think it's not yet clear how many mutations, if you can ever get to that point, where the microenvironment doesn't become important. We've not seen that so far. Every place we look, we see that there's so much communication that is going on with the surroundings that the tumor cells have, that it's a very complex combination of interactions between the stem cells and between all the other cell types that exist within the tumor. In the skin alone, there's 65 different cell types, and so just for understanding the normal stem cells and how they're interacting, it's really it's going to keep us busy for a long, long time. The tumor microenvironment is more complex, but it's a different kind of complexity. So I think it's really still open. The other aspect that I'll just mention is some of the work that we've done that in the normal stem cells that show that stem cells can acquire memories of their inflammatory experiences. It's not simply the immune cells that have memory, but the stem cells have memory as well. It really raises the question of what's going on in cancer, and if memory is cumulative, then it really suggests that there could be all sorts of changes that are happening at the epigenetic level that can be unleashed in certain microenvironmental contexts that we're not even paying attention to right now. So I think, again, some of the recent studies that have been done along these lines are going to be interesting to see how they unfold in the future.
Okay. Dr. McAllister, do you think microbes could be this pan-cancer features that can be used to treat or diagnose the cancer?
So certainly I think they're going to be important, and there's already been some work done on this area, but I don't know. I think it's too complex to think that it, by itself is going to be relevant, and I think it's going to be part potentially of other mechanisms and other signalings, and I don't think on its own. I'm talking about diagnosis. Now for treatment, I do think that, and again, a lot of trials results are going to be coming up this year. We have very little results so far, but I do think there's going to be more and more emphasis on microbes, mostly as a part of treatment, right? So it's just like we have radiation therapy and we have chemotherapy and immunotherapy, microbial modulation may play a part of these treatments, and not only for treatment of cancer in terms of killing the cancer cell, but also for decreasing toxicity in the context of specific diets, to decrease the chances of recurrence. So I think there's going to be a lot of work on that. But again, I feel like the field, of course, we're always getting, as clinicians, anxious to get things into practice, but we still have to learn so much more about the role of microbes in cancer, in the microenvironment. So.
Right. The study on microbiome is just starting. There are a lot of unknown things remain to be elucidated.
Yeah.
Thank you. Yeah.
Thanks, Florencia. I have sort of a related question that I think the three of you all touched upon on that from your earlier answer. So it's essentially the idea of the plasticity, right? Like we've talked a lot about the heterogeneity, the complexity, and the multidimension. There's also the component of temporal changes, like within the cancer cells and within the environment, as Lynn was talking about, the cancer stem cell was reacting to the environment very differently, when they're educated, so to speak, or not. So I'm curious of that dimension of plasticity, or one could say, complexity. How has that affected your thinking of your research question, your research angle, and how do you, maybe a related question is, how do you manage that complexity in the context of both preclinical models and the complexity of sort of using human samples to support the findings? Xiang, you want to kick off?
Sure. That's again a great question. In metastasis, the classic model is genetic selection, right? It's the clones that have the enabling feature that can make it to the next step got selected for, and as a result, the final clone of the successful metastasize is a tiny fraction among the original bulk of tumor. But that was changed, sort of modified by finding that EMT can also bring stem cell features. Therefore, a non-stem-like cell can acquire stemness during time and in response to environment, right? So, and along that direction, you may think that there's a combination, this evolution of metastasis, combination between changes of a single cell and genetic selection. So it's a sum of the two totally independent related process. Therefore, that completely should completely alter our thinking of this whole cascade. Right? It's not necessarily the minority that makes the final step is the winner. Right? Rather, there's a lot of opportunity. There are a lot of alterations during the cell's progression that change their phenotype, change their plasticity. And another perhaps interesting way of thinking is, although cells of different tissue of origin, they are fully differentiated as a normal cell. When they become cancer, they are all kind of differentiated a little bit to what may be a convergent, some convergent feature, call some convergent feature. So I think that's a very interesting question. To handle that, we do need to have some real-time technology to kind of follow the evolution of a single cell at a single cell resolution over time. I think that's something that's lacking. We have a lot of high content, high throughput technology today that can only provide you a snapshot of the tissue sample. But we do also need a way to get longitudinal data, the true longitudinal data, following each clone of cancer cell, each individual cancer cell. I think that ultimately that will provide a lot more insights, deeper insights into this question.
Thank you, Florencia. I'm curious of your thoughts, particularly in the pancreatic cancer context.
Yeah, well, I mean, certainly in pancreatic cancer, I mean, well, first of all, even though I'm an oncologist, I trained in a developmental biologist lab. So everything I can think of, it's, temporal fashion and spatial conformation. So I do think it's extremely important. And I am very conscious about this. And that's why even within the lab, we look with very different tools and very different kind of methodologies at different time points. So when we are talking about early initiation of tumors and early progression, we are kind of thinking very differently than when we have established tumors. So in other words, but I think what happened is it's very clear in the lab, but then when it comes to clinic, I think many times we're applying some concepts that we learned in the early, and we're applying them to patients, and then we realize it doesn't work. Well, when we come back to the modeling, we realize that sometimes we stay with certain models because they're just more sort of homogeneous, right? And as all the models, they're just easier to interpret. But the truth is, it's very, very important. And from microbes' perspective, we see huge differences in terms of patterns, even of distribution, as I mentioned before, at different time points. We are seeing interactions with different even type of cells. And I do envision there's going to be different functions also along that trajectory, from the very early versus when there's a cell already established with several mutations and more committed to a phenotype. So, yeah, I think it's extremely, extremely important. But that's why, even though I'm an oncologist, I'm really focused on early detection and all that, because I think there's going to be a lot easier to prevent pancreatic cancer than to cure it. So.
Excellent point. Elaine, your thoughts?
Yeah, I mean, certainly when Shinya Yamanaka showed that you could start with a skin fibroblast and go all the way down to an induced pluripotent stem cell, all bets are off in terms of what goes on in cancer development and how things dynamically can change. And so, so it's not just the stem cell, and if things are left for too long, then certainly other cells can certainly respond and become stem-like and vice versa. And so I think all of this is really, I'll echo the notion that preventing and early detection is going to be absolutely critical. The other thing that I think really is underscored there is that combinatorial approaches are going to be absolutely essential. I don't see any future really in the sort of single molecule approach to treating cancer anymore. There's just so many examples where you better hit it with everything you've got because things are so dynamic and changing. And one thing that cancer has told us is that it's a whole lot more devious than anyone, I think, has ever expected that to be. I think if you look back at the last 4 decades or 5 decades, it's just, we're not there. And despite all the excitement and all the new drugs, we're still not there.
Okay, treating cancer is a really sophisticated task. I would like to continue this discussion, but our time seems to be very limited. So let's wrap it up here. And it's really late for Dr. Fuchs, and she needs to get offline and take a rest. Let's all join me and thank Dr. Elaine for her great talk and for joining us. Yeah.
Thanks very much.
Thank you.
Thank you.
Wish I could stay on forever.
Hope next time you can visit on site.
So, good night all, and good morning all.
Oh, yeah. Thank you. Okay.
Thank you. Thank you for all the panelists for the really interesting discussion. And as my co-host was saying, we could go on. I think there's a lot to discuss in this context. But we do have 2 very exciting talks coming. So with that, now I'd like to welcome our next speaker, Dr. Florencia McAllister. Dr. McAllister is an associate professor at the University of Texas MD Anderson Cancer Center, and I was just mentioning she has done exceptional work in the context of both basic science research and clinical care in the context of pancreatic cancer. So her talk title today is Pancreatic Cancer, Microbes and Their Induced Traps. Florencia, please.
Thank you so much. Can you hear me well? And it's a slide. Okay. Thank you so much. This is great pleasure for me. So I'm going to give a talk which is going to be basic and then going a bit into some translational angles, although without getting too clinical. So I'm going to start by saying I have no financial relationships to disclose. And I'd like to tell sort of my road to microbes, because being an oncologist, why did I end up studying microbes in this context? So first of all, when I was a postdoc in the laboratory of Steve Leach during my fellowship in oncology, I realized how hard it was to treat pancreatic cancer. So really early on, I realized the best will be to prevent this really aggressive disease. So what I realized it was that most of the risk factors for pancreatic cancer, like cigarette smoking or diabetes and obesity, are all linked to development of a type of immune response, which is led by Th17 cells, which is a type of CD4 T cell that has the capacity of secreting IL-17, a cytokine that in different contexts has been shown to be pro-inflammatory, and in the context of cancer has been mostly studied within colon cancer at that time. It has been shown to be mostly protumorigenic. So I initially started by targeting this type of cell and see if we target this type of cell and we block the production of Th17 cells, can we actually decrease or delay the initiation of pancreatic cancer. And indeed, that was what we published in 2014, that if we use neutralization antibodies against IL-17, which are actually used in clinic for psoriasis treatment, they can actually delay tumorigenesis. And then a second group using a similar model have shown also through a different type of antibody, but in a different type of pancreatic cancer model, but again, the same, that blocking IL-17 can also delay initiation of pancreatic cancer. So when I first started my lab, I went into study deeper, why could this be happening? And what we did is we basically we did a GFP sorting of the panning cells, which are the lesions that are preceding pancreatic cancer. And we did microarrays, and we looked at transcriptomic changes between pannings that were facing neutralization of IL-17 versus pannings that were growing in the absence of IL-17 neutralization. And we looked at changes, and we were really expecting changes in terms of immune crosstalk. We were expecting changes in cytokines, or perhaps that had to do with chemotaxis or migration of cells. But instead, the top signature had to do with embryonic stem cells activation, in particular with two genes that were BCLK1 and POU2F3, which have been described as being present in tumor-initiating cells within pancreatic cancer. So this was very interesting because it certainly made us think that not only immune cells may be interacting with epithelium, but they can also not only be secreting molecules and have sort of paracrine effect, but they may also be altering their structure and their plasticity. Following that, it came this question, which really was posed by one of the reviewers of a paper, or one of our papers, which is why do we have IL-17 or why do we have Th17 cells within tumors? What is the driver? So really to answer that question is when we got very interested about understanding more about microbes. And that's mainly because I was working in Hopkins with an infectious disease and microbiologist, Cindy Sears, who went to show that bacteria, specifically ETBF, can trigger tumorigenesis in the colon in particular through the induction of these Th17 cells. And basically, if you use the APC in mouse model and you add ETBF, you can see a lot of these tumors starting. And then if you block IL-17 with the very similar antibodies that we use in pancreatic cancer, you can actually delay that formation of those polyps. So the fact that an immune response Th17 mediated could be the link between bacteria and tumorigenesis was very interesting to me. So that is kind of putting this scheme very nicely. The idea will be that bacteria, when it gets translocated through the gut epithelium and gets to activate antigen-presenting cells, in turn, those cells are going to activate T cells to produce IL-17. And those cells are Th17 cells if they are CD4, or gamma delta T-IL-17 if they are gamma delta, or innate lymphoid cells. Those three types of cells can have the capacity of producing IL-17 and other associated cytokines. And what Cindy and others like Michael Carney have shown is that those cells are very important locally within the colon for the development of cancer. But what we postulated is that those cells can also circulate systemically and promote other tumors distantly from the colon. So around that time, a lot of work was happening in 3 different laboratories, and these 3 papers came out in 2018, almost at the same time, to show the importance of the gut microbiome in pancreatic tumorigenesis. So in the first work that was published by the laboratory of George Miller in NYU, they showed using the KPC mouse model that if it is re-derived in a germ-free environment, there's a complete delay in the formation of premalignant lesions within the pancreas. And that effect is totally re-skewed when the mice are transplanted with feces coming from a mouse with pancreatic cancer. Very similar results were found by the group of Chris Jobin in the University of Florida. Using a different mouse model, they also were able to show that germ-free environment delays tumorigenesis. And then the laboratory of Ashok Saluja and with postdoc Sethi, they showed in different models, this is in implantable mouse models, that subcutaneous implantation of KPC cells, which are actually derived from very similar mouse model used by George Miller, but implanted subcutaneously, in the presence of antibiotics, which is another way to deplete microbes, they're capable of delay tumor growth. And the interesting thing was if the mice that are recipient of these tumors are RAG1 knockout mice that they cannot get activated T cells to function, then in that case antibiotics are not effective, which means that the effect of bacteria in triggering tumor growth is mediated by T cells. So that brought us to this point of when we were evaluating all of this work and the potential role of the gut microbes in distant tumors. We came across this publication, but also others that I mentioned in a moment, which actually describe the fact that we tend to think about the gut when we think about microbes, but the reality is that microbes are everywhere. Every surface that is open into the outside, any mucosa that has any exposure, even under pathological conditions, can potentially be colonized by microbes. So that brings me to this point, which is bacteria is not only located in the gut, and it actually can be found in very, very rare, unexpected places. And the first description actually of microbes within tumors was given by William Russell. I found that he actually described in the 1890s in this manuscript the presence of microorganisms within breast cancer that then later on were really not studied further for more than 100 years, but with the NIH really getting deeper into the microbiome sequencing and the microbiome human project, there were a lot more papers coming out showing that other sites like carotid plaques, like bladder cancer, and even lung cancer, which again we tend to think of it as a sort of sterile organ, they are colonized by bacteria. So this was of huge interest around this time, and this is when the laboratory of Ravid Strossman in Israel has shown that not only the tumors that I just mentioned have microbes, but basically he sequenced across several tumor types and found microbes across all of them. Of course, in different type of amounts, but even the brain tumors, like you can see here, the GBM, they also contain bacteria. And this was done by analyzing 16S in bacteria. When it comes to pancreas, he published a paper in 2017, which was the first paper to show the presence of bacteria within pancreatic cancer. Not only he showed that bacteria are present in the pancreas, but he also showed that they are important in the resistance to chemotherapy, which is actually gemcitabine, the most commonly used chemotherapy for patients with pancreatic cancer. You can see here in this model that he used when he added gemcitabine on the tissue and he measured caspase as a marker of apoptosis. There was hardly any apoptosis. The same thing if he was only using ampicillin, but if he was adding ampicillin to gemcitabine, there was a huge increase in apoptosis, and the same thing when he added chloramphenicol, suggesting that there is some type of bacteria that increases that resistance to the chemotherapy. And indeed, he went on to show that there's an enzyme, which is the cytidine deaminase, secreted actually by the bacteria, which is capable of metabolizing gemcitabine. So he went on and deleted specifically this enzyme from the bacteria, and he was able to show that in that context, the cells are becoming or are gaining sensitivity to gemcitabine. So really interesting work. But most of this was conducted within in vitro systems. So the question that we asked around that time, and mostly because we wanted to understand from a human perspective, does tumor microbiome matter, mostly from a pancreatic cancer perspective. Around that time, a lot of work came out from the laboratory of John Wargo and Lorenzit Vogel and Dr. Gakiewski from Chicago, all of them showing that there were clear differences in the microbiome of patients that respond or do not respond to immunotherapy or chemotherapy. But we wanted to understand if there were differences in the tumor microbiome between patients with pancreatic cancer. Now, we don't have responders and non-responders to this bad disease. So we can't really stratify patients based on therapy responses. However, there are very few patients who go on to live long-term after surgery for pancreatic cancer, and it's really rare. It's a few patients, as you can see. The median survival for this cancer at 5 years is 10%. So very few patients live more than 5 to 10 years, which is considered the long-term survivor. But what we thought is if tumor microbiome is important, there should be some differences on those very few patients who are at the very benign side of the spectrum in this cancer. And that is what we did. We actually looked at two different cohorts, one from MD Anderson, one from Johns Hopkins, and we matched patients that went to live long-term with others that lived short-term, around a year for the short-term and around 10 years for the long-term survivors. And they were matched for all characteristics, including the stage, age, and other treatments that they have received, including genetics, backgrounds, etc. So the only difference was the survival. And we analyzed mostly the 16S on the tissue, and we also went to do immunoprofiling. And we used the Hopkins cohort as a validation cohort. So what we did find was very interesting. Similar to what was previously found in the gut, higher diversity, but this time within the tumor, was correlated with better outcomes. So the long-term survivors had higher diversity of bacteria. What that means is there's just more heterogeneity of bacteria within the tumors of those patients. And the same thing was detected in Hopkins using 3 different indicators of diversity. More than that, what we did then was to look at beta diversity, that it's a way to look at the differences between bacteria between different samples rather than within the sample itself. And you can see that if there was a very clear differential clustering between the long-terms in blue and the red and the short-term survivors in red in both cohorts. Now, even more interesting, when we tried to look at them together, there were no different clear enrichment of similar bacteria within the short-term survivors from both institutions. However, the long-term survivors from both institutions clustered together, suggesting potential common bacteria driving that long-term survivorship. And when we looked specifically what could those bacteria be, we found that Pseudoxanthomona, Saccharopolyspora, and Streptomyces might be some of those players, and those were the 3 that they were at the top enriched in long-term survivors of both places. Certainly, these will have to be validated prospectively in larger cohorts of patients that are undergoing surgery. The next question we ask is, okay, we found these changes, but are these static changes? Can we actually change the bacteria? Because if we have different bacteria in patients that do better, can we potentially alter that? So how can we do this? To understand where that bacteria is coming from, we actually set up a very pilot experiment, which was really done by collaborating with our surgeons and basically patients that were going to the OR. What we did is collected stools of them in the same morning of the surgery. We took tissue from the Ripple surgery that was confirmed to be tumor and adjacent tissue that was confirmed to be normal. And we did 16S in those 3 samples from the same patient. And then what we did is we actually graphed based on the origin of the microbes. Of course, all of this based on the 16S data. And if you look at the third group, the most interesting thing was in green, you can see around 25% of the bacteria that is found in the tumor is actually coming from the gut. It's not present on the adjacent area. Around 25% is the same bacteria that is also found in an adjacent area. And then there's like another 50%, which is not coming from the gut, and it's not present in the normal adjacent tissue. And likely that might be coming from more like upper GI tract. That's why we cannot see it in those 2. But this certainly suggests the fact that if we want to change the tumor, we can actually change the gut microbiome and expect that there's going to be crosstalk and that some of those bacteria are going to translocate into the tumor. So that led us to experiments that we called generation of what is the microbial humanized mouse that I'm going to mention from now on. And to build this system, we used specimens coming from 3 types of patients. First, what we call the short-term survivors. Those are patients with pancreatic cancer. Most of them had metastatic cancer. The long-term survivors, we call them NED because certainly these are not the same patients from our previous experiment that I showed because, of course, those patients were long-term survivors and they had cancer when they had the surgery, but right now they have no evidence of disease. So this is basically 10 years after their disease was operated. And then finally, we had the healthy controls. So we took stools from these 3 types of patients and basically put it into mice that have received antibiotics for normalization of their microbiota. And we were able to show that if we implant tumors in these mice after the differential colonization, we are able to initiate and maintain colonization differential between these three groups of mice, in which the only difference is the type of donors of stools that they got through fecal microbial transplants. And not only within the gut, but also within the tumor, we were able to find differences between the groups of mice that were colonized with a different type of donors. So with this colonization sort of answer clear, we went into the next question, which was, can gut microbiome modulation affect the tumor immune activation status? And that was very interesting because in our initial study, we did find that long-term survivors had more immune activation, more higher number of cytotoxic T cells, and lower number of T regulatory cells. So to look at this, we performed immunoprofiling at 5 weeks after the implantation of the tumors, and indeed we found that the mice that received the long-term survivor stools had the higher number of cytotoxic T cells and the lower number of immune suppressive cells. And the opposite happened with the mice that received the stools from the patients with pancreatic cancer. So that brought us to the last point, which was, can we actually alter the tumor growth by only changing the gut microbiome? I expect a gut-to-tumor microbial transfer. And indeed, we found that we can do that. And what we found was not really what we were initially expecting, but now we understand that that is certainly, there are a lot of reasons to now explain this. We found that the long-term survivors were able to induce the smallest tumors, and as I mentioned before, these had the highest number of cytotoxic T cells. But on the other hand, the mice that have received the short-term survivors' stools had the largest tumors, and the healthy controls are sort of in the middle. And what we're realizing now that we have done a lot more experiments is that the healthy controls can really be everywhere because it really depends. There's no such a concept as a healthy control. Patients, depending on the diet they're following or the age, they can have differences in the effects they induce. But the long-term and short-term effects are very sustainable. What we then did is we blocked CD8 T cells to see if this effect was really mediated by T cells. And you can see that in this group, when we block the CD8 T cells, the protective effect is lost, suggesting that this effect is indeed mediated by the CD8 T cells. So with this in hand, and sort of circling back to IL-17, which is sort of what brought me into this bacteria world, we asked this question, is bacteria-driven IL-17 playing a role in pancreatic cancer? I'm talking now about established pancreatic cancer. And what we did to answer that question was this experiment, which was really done at the very beginning. I'm showing it now, but it was done a lot before we did all the human studies. When we take stools from patients with pancreatic cancer and we look at cytokines and we compare that with healthy controls, this is mice that have received different FMT, PDAC versus healthy control. When we looked at several cytokines within the serum, we found that IL-17 is the top cytokine that is driven by PDAC stools put into mice, suggesting, as I started at the beginning, the fact that IL-17 within the gut is driven by certain bacteria, which we are still exploring. It's probably, we know that SFP in animals is a common driver of IL-17. We still do not know in the context of PDAC which one will be that driver. But that IL-17 in turn very likely goes on to modulating systemically tumorigenesis. And what we need to really understand better this mechanism is we took tumors from mice that have received IL-17 neutralization antibodies, in this case in the context of the same experiment I just described with the FMT. And after 2 weeks only of neutralization, we did RNA sequencing, and indeed we found, as expected, that most of pathways that had to do with neutrophils and myeloid cells and other myeloid cells were changing in these tumors from mice that had IL-17 neutralization. So it was expected, because that is the known role of IL-17, that is going to be a driver of myeloid cells. Now, what we did then was try to understand if there could be a potential therapeutic effect, but we found that there was no changes in the tumor growth when we block IL-17. However, when we looked in detail at how the tumor microenvironment sort of reshapes or remodels after blocking IL-17, what we found is a much higher number of CD8 T cells closer to epithelial cells when we block IL-17. And also we saw a decrease on the number of GR1 positive and neutrophils within those mice, which makes sense because, again, IL-17 is a driver of this type of immune cells. So that brought us to the point of, thinking, should we then think about combining IL-17 inhibition with immunotherapy, which is an experiment that I sort of resist doing for a long time because I felt like everyone else was doing it, but I'm happy my postdoc convinced me otherwise. And when we combined IL-17 blockade with anti-PD-1, as you can see here in a subcutaneous mouse model, we saw a tremendous decrease in the tumor growth. And then when we use another model, orthotopic mouse model, also using the KPC cells, we saw an improvement in survival. And finally, when we use a genetic modification in which really the IL-17 receptor is knocked down from the KPC cells, and we add anti-PD-1, you can see in this case how the tumors almost melted on those animals. So as a conclusion, the combination of IL-17 blockade with other immunotherapy and checkpoint blockade might be important for this therapy. Now, we wanted to understand better why are these neutrophils playing this immunosuppressive role. And we really came across this pathway, which is NETosis, reading a book on immunobiology with one of the postdocs that I think is in the conference today, virtually attending. And it's the pathways of NETosis, which has a lot to do with bacteria. Basically, NETs are traps that are released by neutrophil, and it's really a type of cell death. It's almost like the neutrophil before it dies. It releases this decondensed chromatin and also has granular context into the extracellular space. And by doing that, it also sort of physically contains certain type of infections. Now, when this happens in tumors, we hypothesize that this might be also physically blocking the entrance of immune cells into tumors to do their cytotoxic activity. So what we did was we added IL-17 to neutrophils to see if there were more NETs, and we did not find that. If you just add IL-17 to neutrophils, nothing happens. However, if you add IL-17 to cancer cells and then you collect that conditioned media and you add that conditioned media into neutrophils, indeed you're able to see a very significant increase in NETs induction or NETs production or extension within those neutrophils, suggesting that IL-17, not through direct contact, but by interacting with the epithelium, can in turn induce NETosis to happen within the neutrophils. So we looked in patients, and indeed when we took the serum of patients with pancreatic cancer and we added them into neutrophils, we were able to see an increase in NETosis versus the serum of healthy patients. So that means that pathways is certainly relevant in these patients. And then what we finally did is we blocked these pathways using different type of inhibitors. We first tried LY6G, which just basically neutralizes the neutrophils. And we can see an effect which is not that different than if we add anti-PD-1, which is interesting. So the neutrophils itself has a pretty significant effect in decreasing tumor growth. But then when we use the PAD14 knockout, which are mice that do not have the enzyme to produce these NETs, then when the addition of anti-PD-1, we were able to see a very significant decrease in tumor growth, suggesting that there's a phenocopy. The NETs inhibition can actually phenocopy the IL-17 inhibition. So potentially it could be at least one of the mechanisms for IL-17 to promote tumorigenesis. And the final question is that we ask, and this sort of just to give you 2 slides on the translational aspects of this talk, and just because I also have 2 minutes left, we ask how can we modulate the gut-to-tumor microbial axis in patients with cancer? So what translational aspect can we exploit? And I just put it all together so I wouldn't spend so much time, but the framework that we are considering is there's good bacteria and there's bad bacteria, and these 2 populations are very dynamic. And the idea is what you need, there's not going to be only one bacteria that is going to be relevant or one bad bacteria we have to get rid of. Very likely these 2 communities will have an interaction, and all we want is we want at the end of the day that those 2 communities, when they interact, that the weight is heavier on the immune activation rather than on the immunosuppression. And you can see just a few approaches will be antibiotics, which we know in other cancers like melanoma, they have a deleterious effect. However, in pancreatic cancer, they seem to have a positive effect. Single bacterium consortia, I don't think it's going to be very relevant, at least from the data we've seen so far. And then FMT, there has been just very recently 2 science papers showing in other contexts in melanoma that FMT can increase sensitivity to immunotherapies in patients that have lost sensitivity. And then from what I just mentioned, mitosis inhibitors are also being studied right now because these are part of the, it could be induced by some of the protomonogenic bacteria. And certainly anti-IL-17, we have a trial right now that it's going to hopefully be run at MD Anderson with anti-PD-1. And we also have a trial with FMT in pancreatic cancer patients that are going for surgery. So with that I leave you with my conclusions. The PDAC microbial diversity, signature, and immune activation status are predictive of survival. The GAT2 PDAC tumor microbial axis can modulate tumors' immune activation, status, and growth. Th17 cells differentiation, which is triggered by the GAT tumor bacteria, can induce and maintain tumor suppression, at least partially by neutrophils and mitosis. And finally, microbial modulation may represent a novel strategy for cancer therapy. Thank you so much. And with this, I leave you with my lab, which is here. This is my current lab in a retreat we had a couple of months ago. And then I want to thank especially to Yu Zhang and Eric Riquelme, who have done most of the work that I have described today. And they're here. They're not in the lab any longer. Yu Zhang is also now here in my collaborators list. He's now back in China in Suzhou Neurologics Bioscience as a senior director. I'm very proud of him and all the work he's doing. And Eric Riquelme is now an assistant professor in University of Mayor in Chile. So I'm happy to answer any question.
Thank you, Dr. McAllister. That's really a fantastic talk. Now we have 5 minutes for questions. Okay, I will take the privilege and ask you a question. So you showed that the cancer-specific microbiome is actually playing a bad role in this tumor progression, right? So if you kill them, then you actually make it more sensitive to the drugs. So you actually inhibit the tumor growth. But clinically, application of this antibiotics usually make it worse, make the immunotherapy worse. So I'm wondering, how do you view this like seemingly contradictory facts?
Yeah, so actually I have to send you my paper. We have a paper published just a month ago in Cancer Medicine. Mm-hmm. Actually, it's really it's been out for maybe like 2 weeks. We actually looked at more than 400 patients. I should have put that slide. More than 400 patients that had pancreatic cancer at MD Anderson, and we looked at patients that were resectable versus metastatic. In the resectable patients, there were no differences, not beneficial or not deleterious of taking antibiotics. The most important factor was, anything related to the surgery, right? What are the margins? What are the lymph nodes? But for the metastatic patients, actually, we found that there's a beneficial effect by taking antibiotics, in particular on the patients that were taking gemcitabine, that got gemcitabine as first line, which goes in line to what David Strossman has shown. Mm-hmm. And a group from Europe has also shown the same thing a few months ago. So it almost looks like all the literature that we have read for other cancer does not apply for pancreatic cancer. And perhaps that makes sense because, you may not have good bacteria to start with in pancreatic cancer. So, you can never get worse if you already don't have good bacteria there, right?
Mm-hmm. Okay, that's good information. I will read that paper. Thank you. And also another question is, what do you think, how the bacteria-mediated immunity triggers the specific anti-cancer immunity? What do you think of the link between these two? How does it reach this specificity?
Yeah, well, we are working on that. It's really complicated. There are many potential mechanisms, but one of the mechanisms we are very interested in is the molecular mimicry mechanism. And there has been work also from the laboratory of Steve Leach and Vinod Balachandran, who have been, they published in long-term survivors. You can actually find antigens that have kind of microbial characteristics, and that might be one of the explanations for this. I don't think anyone has yet done the ultimate experiments to prove that is the case, but that is one of the potential mechanisms.
Right, but usually the abundance of this kind of microorganisms remains very low in the tumor, right? So do you think that's enough to trigger such a strong, anti-cancer immunity?
Well, we don't know if there are. I mean, there are many possibilities, right? It could be the tumor neoantigens. It could be non-tumoral neoantigens, and that's one of the possibilities that we're working on right now. So we are considering all options, but I think mimicry might be the mechanism. Now, what against what, I don't think we know. If it's going to be, again, a mutated antigen, a non-mutated antigen, if it's going to be, a bacterial or a non-bacterial. But, we think that there might be also products downstream of bacteria. That's kind of the totally opposite, hypothesis, that, it may not have to do, directly with mimicry, and it may have to do, directly with metabolites that are being secreted that are, inducing changes or, or regulation or suppression of different immune cells. And we're looking at both. And I don't think it's going to be one or the other. I think it really depends on the context. And, we are starting by models in which we can induce mimicry. And in that context, certainly mimicry is going to be relevant. But that's because we have a model that is directed towards that. In patients, we have seen, based on the data that we have analyzed, that there are different metabolites. And now that we have looked in the serum, we have some connection between the long-term survivors and specific metabolites. And I think those two mechanisms are my main, players, I believe. But I think there's might be others as well.
Sounds like a lot of exciting things ongoing.
Yes, too much.
A very short and specific question. So, if I understand correctly, for the long-term survivors, in the pancreatic cancer, the microbiota is more complex. So that's then eliciting a more robust immune response. If that's true, then if you look at those tumors, are there more tumor-infiltrating immune cells?
Yes, indeed, exactly. We found a lot more immune cells on those tumors. And prior to that, as I said, Vinod Balachandran has also published that you have more CD8 T cells on those long-term survivors with more, higher quality of the neoantigens. And they're also, we've done spatial location of those cells as well, and we find that they're also a lot closer to the tumor. So there might be some functional barriers that are being removed in that context as well, which may have to do with some immune activation. But how is this happening? Is it the direct effect? Is mediated by metabolites? That's what we are trying to learn right now.
Okay, thank you for your fantastic talk. Let's move on to the next session. I'll hand it over to Jiaying. Thank you.
Thank you, Florencia. Very thought-provoking talk indeed. So now we have our final speaker of the day, Dr. Xiang Zhang, the William T. Butler Endowed Professor at Baylor College of Medicine. As we mentioned briefly earlier, Dr. Zhang made really important contributions in our understanding of metastasis, particularly in the context of bone metastasis and the regulation of tumor immunity. His talk title today is The Evolving Cancer Niche Interactions During Bone Metastasis. Welcome. Xiang, I think you're muted.
Sorry, I'd like to thank the organizer for the invitation, especially Jiaying and Shang. It's really my great pleasure to be able to talk to this platform. So I actually visited Hangzhou a few years ago and have very memorable experience. This is one of the beautiful cities I ever visited, and I really hope I could be there physically. So what I'm going to talk today is about bone metastasis. As I alluded to earlier, we are in the process of trying to dissect the temporal evolution of bone metastasis. I'm going to share with you some of the published work and also some unpublished data. I would like to start with some introduction about what we know about metastasis in the bone. This is quite a sophisticated process which has been very elegantly demonstrated by many labs. It can be summarized as the diagram on this slide, especially for the advanced stage of bone metastasis. It can be summarized in one word, which is a vicious cycle. By calling it vicious cycle, what I mean is that cancer cells through paracrine, can activate the bone resident cells, including osteoblasts, which are the bone-making cells, and also osteoclasts, which are the bone resorption cells. Through activation of these cells, they also activate the osteolysis process, by which osteoclasts dissolve the bone matrix, and this process releases a lot of growth factors buried in the bone matrix, and that reciprocally activate cancer cells. And therefore, there's a paracrine vicious feed-forward loop between different cells that really drive late-stage bone colonization. And the current drugs used in the clinic to slow down this process target osteoclasts, but they cannot cure bone metastasis. This is how bone metastasis looks like in the clinic. You're looking at an X-ray photograph where you can clearly see loss of bones in multiple foci in the pelvis bone. And this is very painful diseases. And as I mentioned, once the metastasis enter this stage, you can imagine multiple pathways got deregulated. And although we can alleviate the symptom, we can never really eradicate the cancer completely. At the same time, we also know at a very early stage, some patients, even after their primary tumors are removed, cancer cells can be detected in their bone marrow. Shown on this slide is an example of cytokeratin positive epithelial cells found in the bone marrow, and the presence of such cells associated with long-term survival of patients and the metastasis recurrence of the patients. And therefore, it's reasonable to hypothesize or to envision that this type of cells is the precursor of the devastating metastasis seen on the right side of this slide. A missing link for the long time is, what's in between? How does one single disseminated tumor cells evolve into such aggressive bone-lytic, osteolytic bone metastasis that eventually kill patients? And this is the focus of the research, part of research at my lab. In the field, many other labs have been trying to understand metastasis dormancy, and we heard about perivascular niche, this terminology from earlier talk from Dr. Fuchs in the context of normal stem cell. In cancer, in fact, the detected tumor cells, disseminated tumor cells, can often be found adjacent to blood vessels in bone marrow and other organs as well. When they are here, it's generally believed that they are dormant when they are surrounding the blood vessels. And there's a crosstalk between cancer cells and endothelial cells that reinforce this dormancy status. Some previous work from my lab and recently joined by a few others start to noticing that when cancer cells exit dormancy, when they begin to proliferate and become a multi-cell micrometastasis, they are embedded in the environment that's enriched with osteogenic cells. By osteogenic cells, what we mean is from mesenchymal stem cells to osteoprogenitor cells and all the way to osteoblasts. So this lineage of cells that are able to generate new bone. So we term it osteogenic cells. In multiple models, we observe this osteogenic niche that harbor proliferating micrometastasis. And in this earlier stage, osteoclasts, the bone-resorbing cells, are still pretty scarce. Later on, they will be recruited, and that will promote the advance of the metastasis to the osteolytic stage. The vicious cycle will start. But this is a pre-vicious cycle phase of metastasis. And we have published a couple of papers characterizing the interaction between osteogenic cells and cancer cells. We discovered that there's a heterotypic adherent junction between these two cell types as well as a gap junction. And these tight cell-cell junctions will lead to signaling events in cancer cell that eventually promote cancer cells to further progress. More recently, we employed some global omics approach, trying to characterize the transcriptomic and epigenomic changes in cancer cells when they are interacting with osteogenic cells. We use multiple technologies, including the RNA-seq from the trap staining in the 3D co-culture, as well as extraction of bone metastasis from the bone environment and perform RPPA, reverse phase protein array, antibody array. We discovered that there's a profound changes in cancer cells. We're using luminal cancer cells, which are ER positive, but interestingly, when they interact with mesenchymal stem cells, they upregulate the basal program, which is mostly triple negative cancer cell feature, and upregulate EMT gene expression. They downregulate estrogen gene, estrogen program, upregulate status 3. And if you look at the enrichment of different pathways, you can also find EZH2 complex upregulation. And actually the very top upregulated pathways chromatin organization, suggesting epigenomic reprogramming event. And this is also supported by protein level data. We can see the downregulation of ER, PR, GATA3, these are luminal signals, and upregulation of several stem cell markers, and some EMT genes. Very interestingly, you can find a hybrid EMT phenotype that while the cells maintain cadherin expression, they also gain vimentin, which is a mesenchymal expression. And there's multiple receptor tyrosine kinases that are upregulated as well. And this is the epigenomic reprogramming. Also, this is evident because even if you use a single cell derived population which presumably is homogeneous at the genetic level, we can still see this change. And not surprisingly, this is regulated by EZH2, and using EZH2 inhibitor, we can reverse the change. At the single cell level, there's an inverse correlation between ER expression and EZH2. And that this and a lot of other data led us to realize that in bone microenvironment, there is an increase of phenotypic plasticity, at least in this ER positive model. Later on, we show this is the same case for other models as well. But in particular, in ER positive model, there's a downregulation of ER, especially for cells that are surrounded or in direct contact with osteogenic cells. And that leads to some heterogeneity in the metastatic lesions. And that also gave rise to resistance to endocrine therapies. And another consequence of this, because of the increase of plasticity and stemness, are surrounded or in direct contact with osteogenic cells, and that leads to some heterogeneity in the metastatic lesions, and that also gave rise to resistance to endocrine therapies. And another consequence of this, because of the increase of plasticity and stemness, we also observe frequent further metastasis from the bone lesions. And this is happening to the ER-positive model and also to ER-negative and other cancer type. In particular, if you inoculate cancer cells through intra-iliac arteries to establish initial bone lesion, and then soon later on you can find frequent metastasis to other organs, including the visceral organs and other bones. And this is further supported by an evolving barcode system where we use a mutant guide RNA focus locus so that Cas9 can mutate its own guide RNA. So over time, the barcode will change, will evolve, that allow the multiple massive parallel lineage tracing. And through this technology and the use of very sophisticated bioinformatics, we were able to delineate the evolutionary history of this metastasis, and clearly at least a substantial proportion of metastasis in other organs are coming from bone. So the conclusion from this is bone microenvironment may promote further metastasis. They're not only allow further metastasis, they actually actively invigorates further metastasis through enhancement of the stem cell features of cancer cell. And this is summarizing this slide. Initially, we have the cancer cell may follow organotropic metastasis as the first wave of metastasis. Some of the cancer cell metastatic seeds will survive better in bone, adapt to the bone microenvironment. And under the influence of bone microenvironment, they got reprogrammed and stemness increase, plasticity increase, and that actually facilitates the second wave of metastasis to multiple other organs with reduced organotropism. And we think this can potentially explain a lot of clinical observations that we could not otherwise understand. So what I just told you are predominantly published data. And from this slide on, I'm going to talk about some ongoing work in the lab that's not been published yet. So as I mentioned, the perivascular niche is likely to be the home for dormant disseminated tumor cells, maybe the first foothold for cancer cell in the bone marrow. And later on, or in parallel, there's other niche, such as osteogenic niche, that promote proliferation. And through yet to be unidentified mechanisms, osteoclasts are recruited, and the entire metastasis enter this osteolytic vicious cycle stage. And, we can further metastasize other organs beyond the bone. The question marks indicate the links that we don't yet understand. We don't know, for instance, what's the relationship between these two different niches, how the vicious cycle is initiated, and perhaps, this early stage metastasis can already further metastasize other organs, in which case we may find other metastasis even prior to the discovery of bone metastasis. In other words, a lot of metastases in other organs are indeed from bone, but we don't realize that. So we are actively searching, looking for answers to these missing links, to fill this missing link. But in the next 8-10 slides, I'm going to focus on this particular question, which is the relationship between these two different microenvironment niches. And do they convert to each other, or do cancer cells migrate from one to another, or they compete for cancer cells? And these are all potential hypotheses. We address this question by using clarity approach that allow us to see through the organ and reconstruct 3D structure of the bone marrow and identify the rare spontaneously metastasized cancer cells within this network. And if you look more closely, you can find the single grain DTCs in the network of vasculature. The blue color is the blood vessel, and the magenta color is mesenchymal stem cell, labeled by NG2, which is the well-known NG2 perivascular pericyte marker. In fact, we actually looked at many different markers, and this one is chosen as a representative picture. And by chance, NG2 also turned out to be a very important subpopulation that we will focus on later on. The reason for that is if you knock out different subset of bone marrow cells, such as NG2 positive periarteriolar mesenchymal stem cells, tier 2 positive endothelial cells, and the leptin receptor positive, another set of mesenchymal stem cell, which is perisinusoidal mesenchymal stem cell. You can observe different effects on spontaneous metastasis to the bone. In particular, this NG2 knockout diminished bone metastasis, whereas other knockout have different effects, although they knock out the cell to the same degree, similar degrees. And we were very encouraged by this result, especially interested in NG2. And the reason for that is we all know bone marrow is complicated. The vasculature there is also complicated. They can be divided into different regions. There's a small artery, we call arteriola, and also there's H subtype, H-type vessels, and L-type vessel, and they all have different impact, different functions in different contexts, and their perivascular cells are different as well. specific for NG2, it is believed to be the parasites that adjacent to H-type vessels. And H-type vessels has recently learned to be important for the coupling between angiogenesis and osteogenesis. And that actually means that the parasites here are usually precursors of osteoblasts, and they can be recruited to the bone remodeling site and to contribute to the new bone generation. And this is very important to us, and we actually confirmed that if you make an injury to the bone through bending, through make a fracture, you can find the recruitment of NG2 positive cells into this region. So clearly, NG2 positive cells migrate to bone repair and remodeling site. If you do a lineage tracing using osteocalcin as the marker for mature osteoblast, you can actually see their substantial contribution of NG2 positive mesenchymal stem cells to osteogenesis after fracture. So they are not the only contributor, but they are one of the important subpopulations. And also, if you now count NG2 using DT, DT-induced ablation of NG2 positive cells, you can compromise bone remodeling both in the homeostasis condition and also in the bone repair condition. So without NG2 positive mesenchymal stem cells, the bone is repaired at much slower rate. So far, I've told you that NG2 positive mesenchymal stem cells seem to be important for metastasis. It's also important for bone repair. So is this the same process? Are these two processes coupling? We decided to do this bold experiment. We injured the bone after tumor resection, primary tumor resection, and we measured how does that will change metastasis. And in fact, the injured bone seemed to be a better home, a better colonization place for metastasis when you drill a hole on the bone or when you bend to fracture the bone, in both conditions. If you direct inject cancer cell into the artery and then drill a hole, you can see the metastases are attracted to the injured sites. So this is a very dirty experiment, admittedly. There's many more potential explanations, including the inflammation at site of injured site. I will come to that point later. And this is another view of this process. If you make a drill, you can see the metastatic cells are enriched at this injured site. So we know that NG2 positive is important for both metastasis and repair. The hypothesis here is that are these two processes coupled, particularly by NG2 positive mesenchymal stem cells. In vitro, if you separate NG2 positive stem cell versus NG2 negative bone marrow derived stem cells, you can find their behavior, their interaction with cancer cells are quite different. If you focus on this screen, you can see green cancer cells very quickly adhere to NG2 positive mesenchymal stem cell and they form colonies. Clearly, NG2 positive stem cells are activating proliferation, and in 2D and also in 3D models. And this is the in vitro setting. They also change the migration of these of cancer cells. If you put them together on upper chamber of a Boyden chamber and using osteogenic signal downstairs, you can see that the cancer cell can co-migrate very quickly, very readily with osteogenic cells. So how does this happen? We know that as per our previous work, I showed this slide before, there's a heterotypic adherent junction between the two cell types, osteogenic cells and cancer cells. At that time, we don't know if this osteogenic cells are NG2 positive or not. So we decided to conditionally knock out NK-adherent only NG2 positive mesenchymal stem cell and see if this process is changed. And clearly the answer is yes, at least partially, if you knock out N-cadherin specifically in NG2 positive cancer cell, you diminish this in vitro phenotype, both proliferation and co-migration tremendously. At the same time, you also phenocopy the depletion of NG2 positive mesenchymal stem cells. So if the repair is slowed down and the metastasis is diminished. So this also helps us distinguish bone repair from a simple inflammation, because we don't think N-cadherin in NG2 positive mesenchymal stem cells plays important roles in inflammation. We actually characterize all the inflammatory components in the system in all the previous experiments, and this one we didn't observe a lot of changes in the immune system. So we think this is distinguishable. This effect we observe is distinguishable from the pure mere inflammation-driven breast cancer colonization. So here is the working hypothesis. Within the dormant disseminated tumor cells, in the resting state, they localize in the perivascular niche very close to a lot of mesenchymal stem cells, including the NG2 positive mesenchymal stem cell. In this niche, mesenchymal stem cells themselves are also resting. They are not activated. They're there, but not doing their job. When there is pathological or homeostatic bone remodeling going on, we think both homeostasis and pathological fracturing can stimulate this process to different degrees. And mesenchymal stem cell in the perivascular niche will sense that signal, and they are activated, and they are moving toward this bone repair site to fulfill their function. And this process may be hijacked by dormant DTCs. In other words, DTC may adhere to mesenchymal stem cells, and that this adherence is mediated by E-cadherin, N-cadherin, heterotypic adherent junctions. And once they arrive in the site of osteogenesis, the cancer cell will have a totally different microenvironment. And there, they can take advantage of the other signaling provided by the osteogenic niche and become more proliferative and progress later to a vicious cycle, the more advanced bone colonization stage. I don't have much time to show all the data. I'm showing you the key pieces of data, but so far, I think all the data seem to suggest this model. They are convergent on supporting this model. The final data slide, just as a side evidence, if you look at a bone, low magnification to high resolution, you can see that the single cancer cell, the single cell usually stay close to endothelial cells, but the multi-cell micrometastasis usually very close contact with osteoryx, both in the metaphysis area and also in the cortical bone where they stay on the endothelial surface. And we're doing more systematically this type of characterization to further support our conclusion. I'd like to stop there by thanking all the people who have done the work, and their names are made both there. Their pictures are shown along the way as well, and also the collaborator at the Baylor College of Medicine, M. Anderson, and other institutes, and also the funding source. I'd like to stop there and like to be happy to take any questions. Thanks.
Thank you, Dr. Zhang, for your amazing results. Now we have like 5 minutes for questions. Here, Yilong. Okay.
Hey, Xiang. This is Yilong.
Hi. No.
Nice to see an old friend. Yes.
Absolutely. Yeah.
Yeah. So I'm very intrigued by your observation that the bone fracture is sufficient to attract the bone metastatic cells. I'm just wondering whether you think this is a unique feature related to bone wound repair,
and whether, for example, in any other tissues like a physical injury, like for example the skin, as Dr. Fuchs has mentioned, would also be having similar observation. Like what is your thoughts on this?
I think, similar observations made in other tissues for sure, but the explanation there was related to inflammation. So that was why we spent a lot of time trying to understand in bone specifically, is what we observe is driven by inflammatory cells or by mesenchymal stem cell, a specific subpopulation of mesenchymal stem cell, and the specific interaction between cancer cell and mesenchymal stem cell. And I think the inflammatory mechanism may be in common between different tissues. For this mesenchymal stem cell specific mechanism, I don't really know. To my knowledge, I'm not aware of other tissues have similar mechanism, but I wouldn't be surprised if there are. Right.
Hi, Dr. Zhang. So you have shown that the bone environment actually invigorates breast cancer cells and then help them to spread to other tissues, right? So I'm wondering, do you think this is a selection effect or this is a reprogramming, because the seed and soil model proposed that, the proper cancer cells can adapt to the proper environment and then they survive, right? And it could be, so what do you think of this?
Right. So that's a very important question that we deliberately trying to figure out during the process of doing this project. The way that we figured it out, we use a single cell derived population of cancer cell. We try to eliminate genetic heterogeneity, and we still see this effect. That's number one piece of evidence. Number two, you can transiently treat the cancer cell that derive from the bone by epigenomic inhibitor in very short-term treatment, and then re-inject them back to the animal. You lose all this results. So suggesting that you don't have to repopulate this. You don't have to maintain the genetic trait. You simple transient treatment can do the job. So we have several other pieces of evidence. We think this is a reversible transient epigenomic mechanism. Yeah.
So you think the non-cancer stem cell, they can be reprogrammed to a stem cell-like state, and then they can spread better. Okay.
Or to be more precise, I think maybe if stemness is not a black and white binary, and if they're continuum, what bone does is to increase this, to increase this the stemness of the cancer cell. Yeah.
So for your bone damage assay, if you use the immunodeficient mouse, would you be able to see a similar effect?
Yeah, that's a great question. I was thinking that we should do that while talking. So we haven't done that. We try very hard to maintain the immune system, but we haven't really think to disable it to see if we see the same thing. But we did do immunoprofiling of the injured site. We tried to see if NG2 depletion is affecting that. We didn't see any effect so far. Yeah.
Okay.
But that's a great suggestion.
Right. That would be an interesting result.
Yeah.
Okay. Yeah. The last question.
Yeah. So if you look at epigenetic reprogramming, obviously is important. But there are probably many enzymes involved. Is there a good target that you would suggest that would block this process?
So far, our top target is EZH2. We think, we have collected a lot of data suggesting EZH2 is the major one, but we do not exclude other ones. We didn't exclude other ones. I think there are other cooperating ones that may be also important. We have been pursuing EZH2 inhibitor so far. Yeah.
Great. Thanks so much.
Okay. Time flies very fast. It's time to wrap it up for today's talks. So it's such a great opportunity for us today to hear so many inspiring and wonderful talks, especially during this COVID-19 pandemic. Cancer ecosystem is an evolving concept. Although we've been very successfully implementing immunotherapy in the clinics, but cancer ecosystem knowledge is still limited. So, but it's a very sophisticated ecosystem with complexity at multiple dimensions. But fortunately, we have novel discoveries and the new concepts keep emerging, like what we have heard today. Hopefully, dissection of the intricate interplay between the various cancer components will help us to develop new clinical tools to better control cancer. Okay, with that, I would like to send our special thanks to Dr. Elaine Fuchs, Dr. Florencia McAllister, and Dr. Xiang Zhang for staying up late with us and giving us a very engaging presentation. And also thanks for all the participants on site and also online for joining us today. Please keep tuned for our next webinar events, and we will see you next time. Bye. Thank you also, Jiaying.
Thank you.
Thank you. Mm-hmm.